and more, we will never ever hurt her. So this lecture is a gentle reminder to all of us to take care of our planet. We are privileged to have Dr. Albert Prem Kumar, who is a guest scientist at the Istanbul University, Turkey, and Stockholm University, Sweden. I now invite Dr. Ajesh TP from the Department of Botany, Thoma College, to welcome the gathering and introduce the speaker to us.
Good afternoon, friends.
Good
Hearty
afternoon.
welcome to the SALVOS seminar series.
Thank you.
As we know, there are 117 participants from various parts of the country, even from Gorakhpur, Agra, Tamil Nadu, and even Kerala.
Oh,
And
thank you. Thank you.
this is an international webinar on novel findings in plant space research. I hope this is a novel topic to most of the people. And our today's resource person, Dr. Albert Prem Kumar, is an eminent scientist and a great researcher in the field of plant biology. Former guest scientist of Istanbul University, Turkey, and Stockholm University, Sweden. He completed his UG and PG from American College, Madurai, and MPhil from St. Joseph College, Trichy, and his PhD from Madurai Kamraj University. And he published around 16 international research publications with high impact in various reputed journals, and he published around 106 research publications in various international seminars, workshops, and webinars. And in today's webinar, we will discuss about the development of space biology with special emphasis to plant research. The origin of International Space Station and how space biologists are sending experimental material from Earth to Space Lab will be discussed in detail. Hope you can enjoy this session. He will also trigger our minds by presenting most important plant science research in space. So now I invite our honorable resource person, Dr. Albert Sir, to this current session of SALVOS seminar series. Sir, please.
Thank you. Thank you so much. Thank you. So let me check the sharing screen. Yes. Yes. Ajesh, you will be in the online, no? All the
Okay,
time.
yeah.
Yes. So are you seeing the screen?
Yes, sir.
Yes. What you are seeing? Can you please?
Good afternoon. Stay healthy.
Now it's slide mode.
Yeah. Now it is presentation mode.
Presentation mode. Are you seeing stay healthy?
Yes, sir. Yes, sir.
Yes, Ajesh, in between, I will talk to you. I will
Okay.
check with you. Thank you so much. Thanks for your nice introduction. Good afternoon, everybody. So it's really, I feel very happy to give you a webinar lecture organized by Thoma College, Kerala. Understand? It's very fantastic. I visited the college website this morning. I read everything. It's a really protective college. Let your light shine before humankind. Fantastic. Thank you so much. So before starting, let me thank few people of this college and principal ma'am. Thanks for your approval to this webinar. And then head of the Department of Commerce and IQAC coordinator, Dr. Rajeev Thomas. Thanks, sir. Thanks for this webinar. And then head of the Department of Biotechnology, Mrs. Elizabeth George. Thanks, madam. Thanks for approving this webinar. And then faculty members, Dr. Manjula and Dr. Anu. And then my special thanks to organizer of this Google Meet. From Turkey, he started sending so much of messages and topic selection. Very enthused, very dynamic, energetic, fantastic. Dr. Rajesh, thanks Rajesh for this invitation and everything. Okay, these four people are the pillars of the Department of Botany in Thoma College. And then let me thank Dr. Ranjini Haridas, the first speaker of your webinar series. So she only introduced me to your college. He is the alumni of your college. Thanks, Dr. Ranjini Haridas. And then once again, I appreciate and congratulate both college as well as the department for this international webinar. So with this positive note, and let me go into this webinar. And my focus is only the UG student, because this college is having UG students and many students are participating. I am seeing many top professors, lecturers from all over the country. They have joined in this webinar, but my focus is UG students so that they can develop their career. So I'm going to present in a simple words, simple terms, very simple presentation, but at the same time, no compromise in content and quality. So simply saying, I'm going to discuss global research in a capsule form. Okay. So I'm going to use many keywords. Okay. Students can take screenshot, or you can email me. I will send this PowerPoint. So taking the keyword, you can develop a research career or research field of interest. You can Google it using these keywords. And then you can go for internship in abroad or within the state or within the uh, country. So you can approach a scientist for internship. So by this, you can customize your higher career in your life. So my intention is, or the outcome or the positive yield of this webinar, it's not be getting the e-certificates. Okay, of course, you can get it at the end. It's not e-certificate, but it's the career building. So if you follow my webinar, if you develop contacts with overseas scientists, you can build your career in an efficient way. Fantastic. So with this positive note, let me motivate the young UG students, their what's that smart brain, motivating or triggering their smart brains, and then following the global research. Fantastic. So if you look into the title of my talk today, you can find two keywords. One is plants, that's botany, study of plants, botany, and space research. So these two keywords, and combining these novel findings. The dictionary meaning of the novel, everybody knows that, but I am refreshing your mind again. There are three meanings, major meanings. Novelty means innovative, novelty means inventive, novelty means originality, original stuffs. So if you combine these three, and then in a single word, you can say novel. So novel findings only I am going to discuss. So much of research in space, but I am going to discuss only the novel, thinking in mind, UG students, in a simplest way I am trying to present it. The first keyword, plants, no need to tell, understand. So in the globe, we have around 250,000 flowering plants. The department itself is the botany department, so there is no need to introduce this botany or plants to you. Everybody is botanists, fantastic, biologists. The space, from the school textbooks, we are studying about the space. Okay, so simply saying, universe, we are in the universe system, and especially we are in the solar system, in the earth, within the earth we are living. So the definition for space, in a simplest way, everything in the universe, beyond the top of the Earth's atmosphere, okay, that is called as space. Okay, fantastic. So coming to the space, the 20th century, the start of the 20th century, is well known or identified for the space race between whom? Between USA and United, what's that, Soviet Union. So this Soviet Union is now no more. So that means it's international stuffs, international politics also in this study. So Space research is having 9 major research fields. Among them, one is life sciences. So we are from life sciences, whether botany, zoology, micro, whatever it may be, life sciences. So in those times, the start of the 20th century, life scientists in the world, they had a very big question. Is it possible to have a life in space? So they started trying. If you have time after this webinar, please give a glance on animals in space. When we obtained independence, freedom, at the time, those scientists all over the world, they started sending animals to the space. Animals in space. So in this YouTube, if you type 10 animals that went to space, you can find what are the animals. First cat, first dog, first chimpanzee. So 10 animals they sent to space. So out of many examples, I'm going to discuss only 4 historic examples which are related to our topic today. The first one is in 1957, Soviet Union sent one spacecraft named Sputnik number 2. In this craft, a dog Laika, even in the school textbook, 6th standard, you can read this. Laika, they sent this Laika. The moment it entered into the space, the cooling system was failed. Then this dog died. Then after 162 days, it returned back to the earth. But while returning, it burned up. Then nothing was there. But sentimentally, Romanian government, so they came up with a stamp. So please do watch this YouTube. Small short YouTube, animated and mix a real and animated one mixed together. You can follow it easily. Okay. Then after that, in 1961, America. See the race between Union and America, United States and Soviet Union. So one after another. So in 1961, January, you note down the month January. So they sent one spacecraft called Mercury Redstone number 2. So it's a series of redstones you can find in the Google or YouTube Mercury. In this Redstone spacecraft, they send one chimpanzee, very small and tender chimpanzee named Ham. So they send Ham. So this Ham in Redstone, it's circulated the space Earth for 16 minutes. After 16 minutes, it returned back. It landed in Atlantic Ocean. Then they took back, and then when they open, you can please do watch this YouTube. It's really cute video. I can say it's smartly looked around. So then scientists realized that, space biologists realized that, okay, life in space is possible because the first experiment was collapsed, Laika died, but Ham is alive. Fantastic. So when these people, they did in January, immediately after 3 months, the Soviet Union made a global landmark invention. So they sent one spacecraft called Vostok number 1. In that, they sent the first human in space. In the race, Union won, but now it's no more. You understand, try to understand. Okay, so that man name is Yuri Gagarin. He is the first man. He went to the space from Soviet Union. Fantastic. So if you watch this YouTube video, you can find this. But the main point here is he spent 108 minutes. So the previous one, 16 minutes, Ham spent 16 minutes, but this man spent 108 minutes in space. So 1 hour, 48 minutes. Okay, then France, after 2 years, France, what they did? They sent one animal, cat, first cat in space. 1963, France sent it. And that name, it's a Felicite. That cat name is Felicite. Felicite means Felicita is a good thing. 1963. And in this cat, they embedded a lot of electrodes in the brain directly. So please do watch this YouTube. Really, you can find the real video images. So then the cat went up and it spent some time in space, and in return back, it was alive. So that means life in space is 100% possible. So if you have time, just download this book, Space and Life, and give you a glance. It's a fantastic books. Only by pictures you can understand a lot of things. Fantastic. Life in space. Then after life in space is confirmed, so five major countries, five big bosses. So they join together, five space agencies join together. They are, what's it? NASA, United States of America, Russia, Japan, Europe, Canada, all the space agencies. Okay, Canadian Space Agency, European Space Agency. So all they joined together and they launched one space lab. So as we are having lab in Thoma College, there is a lab in the space. So they send this lab, single lab, to the space. That space lab, everybody knows that International Space Station, ISS. Fantastic. So it's a so coming to this lab is called as floating lab. It floats in the space. Just imagine a lab which is floating in the space, also known as orbital stations or orbital lab, because it circles the earth. It is not, it cannot stand while, understand, it will fly off, no gravity, everybody knows that. So it's called as orbital stations, sky lab, floating lab, as it circles the earth. So a few words about this space lab as a scientific point of view. 391 tons physical mass, and then 400 kilometers from the earth, and then 28,000 kilometers per hour. Just imagine. So it rotates this earth. Suppose after this webinar or during this webinar, if you want to locate this ISS, just like speed post, you can track the location of space lab at present at this time. Where is the station is rotating, in which part of this globe? Spot the station.nasa.government. From India, from globe, anywhere else, you can track the spot the station. Fantastic. So coming to the space station, inside the space station, maximum 8 members can stay. So you can do a lot of exercises because you cannot come out, and maximum 6 months you can stay there. So that means scientists can go and come back. So doing experiments, and these people are astronauts, special people. Fantastic. So now the major point here is, though 5 countries send one space lab, as a single country, China, it sent one space station of China to the space. It is called as Tiangong 1. There is a university also, Tiangong University. So in English, it is called a Celestial Palace. So this lab sent by China, it's operated for 7 years. They also did a lot of plant science, all the research, 9 major research area. Okay. Then in this major significant point, 2 first Chinese astronauts, they went up to this Chinese space lab and they did a lot of experiments. Yang and Yaping, 2 women scientists, women astronauts, they went to the space lab of China. And then in April 2018, China took back this. There is a point called Point Nemo. That Nemo means no man. So it is otherwise called as space symmetry. So what China did, so in April 2018, so it made this lab as a dead lab and took back and put it in this cemetery. Fantastic. Then it sent another lab, space lab. Just imagine 5 countries together, China alone. So I'm not praising, just I'm telling the facts. It sent this second lab. This is the second lab, again Celestial Palace number 2. Again, Chinese, what they did, only 2 years, 10 months, 5 days, it was operational. So last July, they took back, okay? But they announced that a permanent station space lab will be launched in 2022. This is the space history. So now a major question, is it possible for the China to send this space lab just next year? Just only 2 years. Because of this COVID, so we don't bother about this international politics. Listen. So coming to the space. So in space lab, the main thing is weightlessness. At the beginning, scientists, they said it's the gravity is zero gravity. What is zero gravity? No gravity. Later they realized that little invisible minute gravity is there, that is called as microgravity. So if you Google microgravity, you can find all the research articles in this microgravity. Fantastic. So when you have time, please do visit NASA website. In the NASA website, please go through research and technology. So as a UG student or PG student, you should understand what are the research, what are the technology they are having in this space station. And if you are more smart, you can download two Excel files, you can download it. Like frequently asked questions, they have frequently asked reports and data. All experiments report, you can have a glance on it. And all facilities report, like our lab facilities. What are the lab facilities they are having in the space lab? You can easily go through that. So I downloaded this, I checked as on today. 2050 experiments they performed. These 2050 experiments included all the 9 research areas, not only life sciences, astrophysics, astrochemistry. So much of things they discuss, even artificial intelligence. And you can find the principal investigator name. Once you know the name, you can Google it, you can visit their college or university or website, you can visit and then take their email ID. Just to send an email, sir, I read your article, I'm very much interested in joining in your lab. Can I come for 1 month internship, something like that. And many students in abroad, even in Turkey, I saw many students, Indian students, they are doing internship. So if you are having smartphone, that is not important. You have to be more smart. Okay. Coming to the lab facilities, until today, they are having 159 lab facilities. So we have in our lab, physical lab, we have this microscopy and then spectrophotometer, colorimeter. Okay, like that, they have special equipments for to carry out all the 9 cycle. What's that? 9 major research area. You can find wet lab, RNA, smart cycle facility. So likewise, they are having it. So these are all the lab facilities. So coming to the botany. So in botany, we are studying very simple study, plant morphology and anatomy. And then also we are studying biotechnology and tissue culture. And then more sophisticated and advanced and modern study plant genetic engineering. So almost all aspects of plant science studies were already done in space lab also. So fantastic. It's a really fascinating, really more interesting research. I can say that. So for today's Google Meet, I am going to discuss only 7 points because 40 to 45 minutes is the best time to present a webinar. So 7 points I am going to discuss. So we will start point number 1. Sending samples. How they are sending samples? From from Malappuram to Kochi, you can send the samples by ice bucket. A student can carry, they can do. But from Earth to space, how they are sending? So how? That is the major question. Very interesting question. And you yourself imagine as a international space station. You are the lab. So postman is bringing. So how they are sending Earth to space lab? So postman, sometimes he used to bring postal letters to our home. Understand? So with this example, I'm going to demonstrate. The postman is payload. I'm using technical word. First technical word is International Space Station. The second one is the delivering person. That is payload in space, payload. Understand? And what he is carrying are experimental samples. So International Space Station, payloads, experimental samples, and what he is using as a vehicle to go to this home. It's orbiters or shuttles. 4 keywords. If you understand these 4 keywords, so you can easily follow how they are sending samples. Okay, fantastic. Already I discussed about ISS. It is rotating. And coming to the experimental samples, don't think that it's only plant samples. It can be microbial samples. It can be animal samples, it can be mammal samples, and it can be plant samples. Almost all experiments, understand? So even you can have what's that, space microbiology, animal research in space, human research in space, so much, so much. If you are really interested, you can email me. I will send you a link for a book to download free of cost. No need to spend even a single naya paisa. Download it. Just go through that. Then you will develop a lot of ideas. So I am from the oceanic aspects. I am going to discuss only 7 drops or 7 points in this. The first point is sending samples, so experimental samples. Coming to the postman payloads. So two kind of payloads they are using to send to space. The first one is STSS. Second one is SpaceX Dragons. STSS SpaceX Dragons. Okay, fantastic. So coming to the STS, yes, like Kerala Transportation Corporation, they have space transportation systems. In Wikipedia, you can find what are the missions they send. So from 1981 to for 30 years, they use this STS. Okay, this is the structure of STS. And until 2011, they used this. For 30 years, they used STS. And you can find the number STS1 is the first one. The STS135 is the last one. 30 years they used. Okay. In STS1, there is no experiment at all. They just send, tested whether it is going and coming, tested. Fantastic. And then in this STS, so Pride of India, Kalpana Chawla, she involved in few STS missions. In STS 107 only, she passed away due to a disaster. So this is Pride of India. Then another Indian women, Indian origin women. So I am encouraging all the women participants, women students in this webinar. Please develop your career. Understand? So the world is in your hand. So you have to do that. So in STS 116, another Indian woman. So origin Sunita Williams, she also involved in it. Okay. So after STSS, the American NASA, they decided to hand over to private company called SpaceX company. So if please go through this website, you can find more information what I am sharing here. Understand? So from 2000, what's the 2012 onwards, they started this. Okay. So very recently, this SpaceX company, they sent this Falcon 9, probably you would have read it in news or in the television. So they sent 2 people to the space. Okay. And this is the news information. So at the end of May, it went to this, it joined or docking. What is that? Docking with the ISS lab, the SpaceX Falcon 9. And then last Sunday, 3 days ago, these astronauts returned back. So SpaceX. Then what is SpaceX? So STS is another vehicle. This is also another vehicle. Understand? So this delivering thing, it's you can see like a bullet-like structure, very elegant in structural design. Okay. So then coming to the SpaceX Dragons, CRS-1, they named us as CRS-1 from 2012. And the last one is CRS-20. 4 months ago, they launched this. So CRS-1 to CRS-20, instead of STS, they are sending samples. Okay, fantastic. So now the next one is in 30th October 2020, CRS-21 will be launched. So if you are really mindful of this, you can follow this. Fantastic. So then you may wonder why they changed the STS structure to this CRS structure. The main point is, the main comparison is, the STS 1 or 2 tons, whereas CRS just 4.2 tons. That means, so they reduced the shape and size and they used light material, but everything is same. Everything is same. 8 astronauts can go, understand? So they can go to this and these are all the things. So 24.4x times weight reduction. So simplicity, simplified version, elegant in structure, looking beautiful, very lightweighted. And then coming to the last point of the first thing, what's that? Orbiters. What is orbiter? So which one carries this payload and samples? There are 5 orbiters: Columbia, Challenger, Discovery, Atlantis, and Endeavour. Okay. So sometimes they will send this payload which carries the samples to the space lab, to Columbia, and then it will come back after delivering. It will come back. Then they will to pick up. They will send another one, Atlantis or Discovery. So it's a random choices, depend upon the experimental design and the speed and everything. There are so much of things. The second point is 7 point. So the second point is performers. You may wonder whether all scientists can go to the space lab. No, nobody can go to the space lab. Then who will perform experiments? So if you follow this experimental flow, you can understand it. This is here. This is the space lab. So all the pre-launching preparations in the Earth, and they will launch the payload and this, what is it, orbiters. It will go to the space station and deliver, and they will perform experiments. After finishing the experiments, they will bring back all the samples to the ground. Post-flight analysis. There are 3 major phases in this experimental flow. If you understand these phases or flow, experimental flow, you can easily understand any research articles published in space biology. Fantastic. So like KFC, there is a KSC also, Kennedy Space Center. Everybody knows that Kennedy Space Center in Florida. So in this Kennedy KSC, 2 major facilities. One is Life Science Support Facility, LSSF, in NASA. At the back of this Life Science Facility, they are having the launching pad, KSC Launch Complex 39. In YouTube, if you type this keyword KSC Launch Complex 39, you can find 1 hour video. You're having a headphone just to listen. It's a fantastic one. How they are launching this and this, everything you will understand. Fantastic. So coming to this again, Kennedy Space Center in the Life Science Center, pre-launching preparation. Suppose if they are sending plant tissue culture samples, so they will prepare the plates and put the seeds and cover this, sterile conditions, everything, and then they will put it in the payload, and then this is called as pre-launching preparations. And also after finishing the experiments, when they come to the ground, they take these samples and then they will send to the principal investigator's lab with extreme care. Fantastic. So these 2 aspects are carrying out in Kennedy Space Center life science facilities. So then coming to the performers. So the performers are astronauts. You see in STS-2, that is in 1982. So very simple experiments they perform. So only 2 astronauts. So slowly they started increasing the number of astronauts and the experiments also. STS-131, you can see 7 astronauts, whereas the last STS-135, 4 astronauts. See, all these astronauts are very brainy and brilliant. They will perform astrophysics experiment one day, and another day they will perform plant biology experiments. So the tasks are assigned to a particular astronauts, and they have direct contact from the space lab to the Earth, the scientists, the principal investigator. Suppose if the scientist is in United Kingdom or in Russia or in these 5 countries, they will talk to them, and in case of any doubts, any problem, they will discuss directly with the scientists. So it's a really fascinating research work. STS-135 in University of Florida, they are having one free course. So if you are really interested, you email me, I will send the link. So you can do a small course in this University of Florida. Fantastic. You can see few pictures. You can see these astronauts are minded in space lab, no weight. So they have to practice in a special way by flowing, and they have to do all these things. Okay. So this astronaut is unpacking the payload. He is opening it, and he is performing experiments carefully. And this women astronauts, you can see how she is balancing. She is using a centrifuge, special centrifuge to use hypergravity. So coming to the third point, so the first point is sending samples. The second one is performance. And I'm going to discuss only 5 novel findings, research findings, what they have done in space. So Professor John Kiss, his name is John Kiss, North Carolina University professor. So his research at the beginning, even his PhD thesis or his lab establishment, so he focused on a field called gravitropic sensitivity. So he checked this amyloplasts. So in the roots, in the root tips, there are plastids, colorless plastids. They are called as amyloplasts. And he studied this plant in a global model plant, Arabidopsis thaliana. And one point I want to tell here, I am expert in Arabidopsis thaliana. If any one of you are interested in having Arabidopsis research in your lab, please contact me. I am ready to help you how to purchase seeds, how to do that. Okay, this John Case, he designed a brilliant experiments in those days in 1989, having a small camera. It's not a digital camera, manual camera with a film. He took 2 ecotypes of Arabidopsis thaliana. In a simplest way, I am telling 2 ecotypes. One is a WT, that is wild type. Another one is starchless mutant. By a mutation, genetic mutation, the Arabidopsis plants mutants which cannot synthesize starch. So in that mutant, no starch at all. So that means he is comparing a plant with starch and a plant with no starch. So when he compared, anatomically he found that in the wild type, in the root tip, in the tip, in amyloplast, dense starch granules, whereas in the case of mutant, no starch at all. So he came up with a hypothesis. Starch is important for gravitropic mechanisms. Okay, so he mentioned starch as gravisensors. So they are sensing. So starch molecules sense the gravitropism. What is gravitropism? Plant growth, plant root growth towards the earth. That is called as gravitropic response. Fantastic. So he came to know that STS-81 in operation in 1997. So what John Kiss did, he repeated the same experiment in the space lab in STS-81. He sent samples. So the payload is STS-81, the orbiter is Atlantis. He sent samples. So you won't believe, so same responses he got it. You can see the original picture of his research. He got the same responses. What is the same responses? In the wild type, roots are growing towards the earth, whereas in the case of starchless mutants, they are growing laterally, not straight roots. Understand? So he come to a conclusion. So starch satolith hypothesis is perfectly fitting even in space also, space microgravity also. So this is the thesis, what's that hypothesis he proposed. So in the root tip, in columnar cells, so the starch grain, starch molecules, you can see a lot of dark iodine spots. These starch molecules, they are having pulling effects, centrifugal effects towards the earth. So that is called as gravitropic responses. Those starch molecules in amyloplasts are called as gravisensors. So this is the excellent novel finding in 1980s. So there's another finding in the start of 2000, a German professor, Rudiger. Why I'm telling professor, you can Google his name and get his email ID, contact him. So University of Tübingen, you can see a small, this is a southern part of Germany. So he sends samples in STS-55. STS-55. So what he did, so protoplast fusion studies he has done. So 1997. So he collected tobacco plants and isolated protoplasts and sent to space, and he checked the protoplast fusions. And the protoplast fusion in plant science, everybody knows that when you have protoplast and due to this aggregation, they fuse together and form a single cell. So protoplast fusion. So in space, during protoplast fusion, what are the biochemical changes are happening? He started studying. So he focused on three aspects of biochemical things. One is an enzyme called peroxidases. This peroxidases is an antioxidative enzyme, so which will remove the antioxidants. It is called as scavenging enzyme. Scavenging enzyme. So oxidative scavenging enzymes. And then another enzyme which is involved in secondary metabolism called phenylalanine ammoniase. And this enzyme also changes as per this microgravity. You can see in mic. Hello, Ajay, can I continue? Hello.
Continue.
Yes. In microgravity, the bands are very dense. So these enzymes are activated in microgravity conditions. And another protein called osmotin. Osmotin means during water stress or drought stress, they are activated. So in 8 hours of what's that? This osmotin is reduced in the case of what is that microgravity conditions, whereas in the case of hypergravity, 1 gram in ground, it is induced. So he found that a definite molecular signature is existing in this tobacco protoplast fusion in space lab. So this is one of the novel finding. He got it. Coming to the third finding, point number 5, 7 points, fifth point. But finding number 3, professor called Jan Lee. So Kansas University professor. So for encouraging girl students, I have selected few women scientists who did a lot of research in space. Okay. Professor Jan in Kansas State University. So she sent samples in STS-81. Okay. So this is the fungal Phytophthora sojae. It causes stem rot disease in soybean. So this is the picture. This is the ground picture. So the stem of soybean becomes red in color. Okay, so this fungal pathogen, so he studied in STS-87 mission. In the same mission, Kalpana Chawla also involved. Okay, so that is the pride of India. So she performed this experiment, and then a novel finding they got it. So this is the structure they used in space. Okay, this is the seed, and then at the bottom they have fungal inoculum. This fungal inoculum slowly goes up, tubing through a capillary force. So the same experiments they performed in ground and space and flight, they compare this. You can see this ground is very healthy, whereas in the case of space flight, the susceptibility increases a lot. Okay, then this is the thing. Then they fixed with glyceraldehyde solution. They took back soybean plant seedlings, and then they analyzed anatomically. They found the plants which are growing in microgravity conditions, they have huge amount of fungal hyphae in the anatomical structures in the cellular regions. So that means microgravity induces fungal susceptibility in soybean. And then they found this conidia and all. You can see so much in their anatomical structures. And then they checked the reason for this. So during this inoculum, ethylene protection is very higher in space flight as compared with the ground. So this could be the reason. So protection of ethylene from this setups, this is the reason. This is the novel finding. Fantastic. Then coming to the sixth point, novel findings 4. So in STS-42, an excellent experiments they performed. That is 2 scientists, one from United Kingdom and another from Scotland. So Professor Maher and Professor Briarty, they perform experiments in STS this. And then they use this European Space Agency biorack. So this is the apparatus they use. So inside the biorack, when you open the biorack in this, they have a growth container, so small box-like container. And then, so this is the thing. So in this box container, they can put these seeds. You can see a white colored, what's that, nutrient filled tissue paper like structures. So if you put the seeds in a sterile conditions, it will start growing. So they check this condition. So nutrients will be injected in this path. So this the box they used it, bioreact glow box for microgravity conditions. So because they cannot keep this container outside, it will fly up. So they have this special European space box. So using the gloves in two sides, they can introduce this container here. Okay. And then they checked four conditions: microgravity, flight 1 gram, flight conditions, 1 gram ground, and 1 gram ground centrifuge. So four experimental conditions they had, they compared. And then they brought the Arabidopsis plants to the Space Life Center. And then they checked with the sophisticated electron transmission microscope. They analyzed the lipid droplets. So you can see in the microgravity, more lipid droplets accumulations. It's a really novel finding. So this professor found that. Then she came to a conclusion. Then in microgravity, the seed reserves, especially lipid utilization, is very delayed as compared with the ground samples. So this is also one of the novel findings. Coming to the sixth one, findings 4. So two collaborations, one from Madrid, Spain scientist, Professor Francisco Xavier from Spain. Okay. So one women scientist from France, Dr. Carole, both together, they sent samples. In 2014, they published this paper. Okay. In STS 133, recently they sent this 9 years ago, they sent samples and then they published papers after 3 years. So they focused on what's that, this rough we know this rough endoplasm and smooth endoplasm, endoplasmic reticulum. So they focused on rough ribosomal proteins. And then with ultracentrifuge, they collected these microsomes. And then in these microsomes, without any ribosomal attachments, you can see smooth ribosomes or smooth microsomes. They collected and then they checked the proteins. What are the proteins are changed in microgravity conditions? They found that proteins associated to auxin metabolism and auxin trafficking, they were totally depleted in these smooth microsomes. Whereas proteins associated to stress responses, defense responses, metabolic changes, they were more abundant. That means they synthesized in higher levels under microgravity in space conditions in smooth microsomes. Then coming to the last point, that is seventh point, finding number 5. A Chinese experiment. Again, Chinese scientists, women scientists, Professor Zheng from Shanghai Institute of Biological Sciences, her email ID and her number also here. Okay, so she sent samples in this what is that Celestial Palace number 1. That spacecraft is called as SZ8. Okay, so in this, so this is the apparatus used in this Celestial Palace number 1. You can see a culture chamber and then a centrifuge. In the Celestial Palace 1, they have these facilities, and you can find here, so callus culture in the space, whereas callus culture in the ground, the number A. Okay. Then she compared the flight ground and the ground control. The proteins only she compared. In coming to the proteins, 65 proteins were upregulated. They are increased in their levels. They are 22% stress proteins and 22% cell wall proteins, whereas 16% intracellular proteins were upregulated and 14% mitochondrial proteins were upregulated. Coming to this, what is that? Again, compared with this, 681 proteins were downregulated. They are decreased in microgravity conditions in space flight conditions. So what are the ribosomal proteins? 20%. Metabolic protein 17%, signaling protein. Signaling itself is a very big domain. Recently, one of the professors asked questions: salicylic acid signaling, jasmonic acid signaling, ethylene signaling, and all these proteins were reduced in space conditions. And then finally, 11% of stress proteins. Why I am discussing this increase and decrease in proteins? Through genetic engineering, you can change this, what is that level of expression. Suppose if it is decreased, you can increase by over expression. If it is highly increased, you can knock down those things. So by genetic engineering, you can play the expression of proteins. Fantastic. The same professor in the same missions as he performed another experiments. In the second project, she compared chloroplast proteins and then mitochondrial protein expression in callus of Arabidopsis thaliana grown in Celestial Palace #1. So the results are here. In coming to the chloroplast proteins as compared with ground samples, 37 proteins were increased as changed, and those 37% are redox proteins. So that means oxidative defense proteins changed 37, and then amino acid biosynthetic proteins 12%, fatty acid metabolic proteins were changed in chloroplast. Coming to the mitochondrials, 50 proteins were differentially expressed, and most of them are what's the TCA cycle. Okay, it's a major respiratory cycle, metabolic metabolism in this, and then primary metabolism, electron transport chain, and then protein synthetic machinery 14% reduced. So with this findings, 5 findings, let me conclude. So what all I discuss is a small seed information. I didn't discuss in detail because the webinar should be a 45 minutes webinar. So with this seed information, you can develop into a tree. You can focus on any paper. If you are really interested in any paper, you can contact me by email. Understand? To students, please, every semester vacation after this COVID time will be easily passed away. So please develop your career path, and every vacation, please join. Don't waste your vacation time. Go for internship, and your biodata, your CV should be unique, unique, unique. If you have this, your career is excellent. Thank you all. So this is my email ID, albertprem@gmail.com. Please note down. If you need this PowerPoint or any clarifications, you can email me. Thank you so much.
Thank you, sir, for this wonderful session.
Sure, Master.
So the floor is now open for discussion. Anyone who wishes to ask any questions can directly ask sir, or can type in the question in the chat box.
No questions? Any questions? You can discuss. Please feel free.
So anyone who is interested to ask any or clear any doubts, you can freely ask, sir.
Whether it is clear or not clear.
Sir, I have a doubt.
Sure,
How can we continue in this career, like Indian space research? How can we continue?
Excellent question. I think
Biology student.
I am seeing Dr. Rajamani from Bangalore. He is watching us. And recently, I want to tell this actually, this space research, as far as India is concerned, they're not doing any research. They are focusing on weather data and education. Okay, that is one thing. Whereas in the case of this NASA and China, these people, they are having a huge money, to be honest, a huge money they are having. So they are doing a lot of research. And recently our director, ISRO director, he made a public request in newspapers. Due to COVID, many companies are not sponsoring. So he invited for startup. Understand? So career as of now, only in few years, an effective government will focus on this career-based space research in India. Clear, ma'am? HOD, ma'am.
Sir, thank you, sir. One doubt from Shinoj Sanu.
Sure,
He's
ma'am.
asking, sir, is there any germination-related studies in space?
Yes. So much, so much of things. How they are germinating. And then this study I presented one, no, the seed reserve utilization, also a germination study. And then two proteins are involved in germination. They are called SPR1 and SK4. These genes are involving. So much of studies, ma.
Okay, sir. Thank you, sir.
Sure, sure.
Dr. Brahim, I want to ask one question.
Sure, sure, sir.
Actually, the work related to stress physiology.
Yes, yes.
Yeah, you said that the jasminic acid and one more acid, what's that? I forget. Salicylic acid.
Salicylic acid.
Yeah, this and all. Actually, when I was working in microbiological challenges with plants, these two substances are very important actually. So it is really very happy because this space research does these two substances, and if our students, whatever they are listening now, if they concentrated these two substances in research area, I think they can find very good solution for stress physiology. I think you can save the plants from stress, and they can improve the crop variety. They can develop improved crops if they focus on these two substances, jasminic acid and salicylic acid. So, Mr. Dr. Brahim has opened a very good window to step up into the area of the salicylic acid and succinic acid. I request all the students, those who are listening to this class, please focus on these two substances in your research. It will save a lot of the farmers' economy. Okay, because nowadays the farmers are facing a lot of problems in stress physiology. Even definitely, you know that there are two areas people are suffering. Farmers particularly, they are suffering. If it is overflooding, also they are suffering. Even drought, also they are suffering. These two natural calamities are making the people to very worst condition nowadays. So better the students, those who are listening this seminar, it will help a lot, and focus on these two substances, how to deal with these two substances of plant physiology. And you can join with Dr. Brahim hands, and please come up with these two locked problems. Please open up and do that experiment with Dr. Brahim Association, and come up with a good result for our society. Thank you very much, Mr. Brahim.
Thank you. Thanks for your suggestion. I saw a few questions in the chat box. Ma'am, HOD ma'am, can you please?
Sir, Renuga Devi ma'am is asking, sir, plants in space, are they being cultivated in vitro condition? If yes, how sterility is maintained?
Yeah, as we are doing in this, now they have sophisticated sterile cultures, and they have very good glow box, which can be like laminar airflow they are having in space station also. Same thing what we are doing in our plant tissue culture, and in fact more advanced equipments they are having it. So 100% sterility they can have. It's a good question, excellent question, I can say. Then I saw another question also, ma'am.
So, yes, sir. Whether long exposure in space can cause mutation in cellular level.
Excellent question, ma'am. Actually, if you watch the pattern of experiments in space, also they are doing in a controlled environment. They are not exposing the Petri plate outside, this and that. Exactly like growth cabinets, they are having the culture chambers. So in space, only by cosmic mutation, there is a possibility of mutation. As and now, they are focusing only on one aspect, that is microgravity. So whether so gravity only exposing long hours or long days of microgravity will not affect the DNA. It will not cause any mutation. They are not lethal. Gravity is just a stress. That's all. Thank you, ma'am. Any other question?
Thank you, sir.
Sure, ma'am. Sure.
Anyone who is having any doubts, you can clear it. Yeah.
Please feel free, discuss. No need of asking questions. You can discuss. It's a chance to discuss. We are overcoming COVID. Please do discuss. And if you are really interested, email me albertprem@gmail.com. I will send the PowerPoint.
So I want that point.
Yes, yes, sure. Okay, ma'am. I think it's a time to wind up.
Everyone is thanking you for the nice presentation.
Ma'am,
One question came.
yes, ma'am. Yes, yes, one
Yes,
question.
sir.
Yes,
I am
100%.
interested.
Yes, ma'am. Yes, I read
Satya
the question, ma'am.
Narayanan. Yes, sir. Okay.
100% possible, ma'am. Whatever background, even see nowadays only we are discussing zoology, botany, this and that. In Western, understand, science is one. So I will give one example. In Harvard Medical School, a lady professor, understand, she is doing plant science research in Harvard Medical School. So interdisciplinary, multidisciplinary, cross-disciplinary, transdisciplinary, all the disciplinary anyone can do. It's a really nice question, ma'am. HOD, ma'am.
So thank you, sir.
Sure, ma'am. Sure, ma'am.
Can you explain about hydroponics in space?
Yes,
Renu Tesneem has
yes,
asked.
ma'am. Yes, excellent question. I am seeing that, and actually I didn't touch the lab facilities in this webinar, because if I touch lab facilities, okay. So if you look into the module, the ISS station module, in each module, the design of the each module is having lab facilities. Okay, in one facility called Veggie chamber or Levada chamber, there are two chambers, and these chambers they are having very special design for hydroponics experiment. Another German scientist, he performed the experiments with using Euglena gracilis, and he found that this what is that flagellae are more important for gravitropic responses. So what we are doing in hydroponics in the plant tissue culture lab can be done in the space also. The same facilities they are having in the space also. Clear, ma'am?
Thank you, sir.
Yes,
Thank you, sir.
yes, yes, ma'am.
So, we shall conclude the session. So I now
Yeah.
invite Dr. Manjula KM of Botany Department, Thoma College, Chungatara, to propose the vote of thanks.
Hello.
Yes, ma'am. Yes, we are listening, ma'am.
Okay. A warm good evening to all. This was the end of the second lecture in the webinar series, SALVOS. I hope all of you enjoyed this section. Now, on behalf of Department of Botany, Marthoma College, and all the participants, I express our sincere gratitude to our chief guest, Dr. Albert Premkumar, for the wonderful lecture on the topic Novel Findings in Plant Space Research. He made this section really interesting and useful by taking us to the fascinating world of this interdisciplinary approach of science. Once again, thank you, sir. Thank you very much for
Thank you, ma'am.
sharing your valuable time and knowledge with us.
It's my pleasure, ma'am. Thank you.
Okay, thank you, sir. Then the entire team of Salvos is also grateful to the participants, including college students, research scholars, teachers, and all the faculties from various institutions who made this section a great success. And also, with great pleasure, I thank the coordinators of this webinar, especially our head, Elizabeth ma'am, Dr. Ajay sir, and Anu miss, and the technical team of our college for their efforts and support. Thank you.
Thank you, ma'am. Once again, on behalf of all the organizers, I extend a very warm note of gratitude to Albert Premkumar sir and all those who have taken part in this session. The
Thank
next
you.
session of Salvo series of academic lecture is scheduled to be held on 12th August 2020, that is next Wednesday. The lecture will be on Bryophytes, a traditional medicinal hub led by Dr. S. Sahaya Satish, Dean, School of Biosciences, St. Joseph's College, Tiruchirappalli. The link for registration to this session will be sent to you by this weekend. We invite you all to participate actively in the third session. Once again, thanking you all. Bye. Stay home, stay safe.
Sure, ma'am. Sure. Bye-bye.
Thank you, sir.
Thank you, sir.